All right. Aloha kako, everyone, for joining us. Thank you. Today is our next installment of Kanaka Genetics. And today we're going to have a very special focus. Many of you will know that it is the day after a very special Aloha Friday and Kamehameha Aloha Friday. So we're spending this Saturday talking about some very new ideas. And we have some incredible speakers to Kauka from your community. And I'm going to start off with some kind of housekeeping arrangements, some details. First, by just thanking all of you for joining us so that we can talk about the health of our community. So let me begin by sharing my screen. We appreciate you guys joining us on Saturday. All right, so the title of this series of two talks, is Emerging Research in Hawaiian Health and Well-Being. We're going to have a sort of state of the field where we'll, talk about two contrasting ideas in genetics and genomics, and we'll have two experts on those subjects with a particular focus on cardiovascular genetics. So heart disease, stroke, and something that is the #1 cause of death, unfortunately, in America. So let's begin by just, again, yesterday was Kamehameha Day, beautiful pictures always. Just wanted to acknowledge that and celebrate that. And of course, a big mahalo nui to all of our supporters. And we wanted to acknowledge Ahahui, Papa Ola Lōkahi, Molokai General Hospital, RCMI, Ola Hawaii, and JABSOM and UH Manoa for all of our partners and the kind of community and lahui that we're building around Hawaiian health and genetics. So also, I should note that we would greatly appreciate it if you fill out your evaluation forms. The majority of this series of talks, and when we get to the questions, will be recorded, and that will be available online in the future. So just be aware of that, and wanted to let you know to be on your best behavior. And mahalo nui for that. And please, again, fill out your evaluations. Your kind of iterative feedback lets us create a better product in the end. And yes, we will be taking questions from multiple platforms, I believe Facebook and other places as well. So we're looking forward to getting to that. And without further ado, introducing our two lovely Manawahine Kauka today. First up, you will have a talk from Dr. Dowsett, and she will be discussing Mendelian disorders, and we'll get to what that means and what monogenic disorders and something that could happen where one gene might have a mutation that could lead to a type of disease. And then Dr. Brown is going to talk to us about common complex diseases. And these are the vast majority of disorders that kind of people develop. These are disorders where you'll have multiple genes that might be kind of contributing to a health state. Just some key terms and variables, some ideas. Again, Mendelian disorders, Mendelian inheritance. Remember the old monk, Gregor Mendel, working there in his monastery with all the pea plants, counting them over and over again. This is where the term comes from. He's the very famous, older generation of geneticists, but he focused mostly on agriculture. And some famous diseases that examples are sickle cell anemia, maybe you've heard of this, cystic fibrosis, thalassemia. These are examples of Mendelian disorders. Dr. Dowsett is going to go in depth on this in a moment. Secondly, we have common complex diseases. These are complex diseases that are caused by a combination of genetic, environmental, and lifestyle factors, and some of which have not been identified yet. Many have. And so we'll talk about how complex that relationship really is. And that's what Dr. Brown will discuss. And then we'll slide into questions. So first up, mahalo nui for joining us, Dr. Dowsett. And you can begin.
Perfect. Hi, thank you so much, Keolu. Let me, I'm going to share my screen, and then let's see if we can get this going. Okay. All right. Are we good to go?
Raja, go for it.
Thank you. All right. Thank you again, Keolu. What a lovely introduction. And again, mahalo piha to all of our groups who are supporting us today. We are again truly grateful for this opportunity. I'm just going to be talking about Mendelian genetics. And again, those monogenic diseases, right? We're talking about, like Keolu mentioned, one gene that then leads to disease that we can kind of, again, diagnose, treat, and manage. So I'll give you a little bit about genetics and diversity, talking about diagnosis making, and then moving on to bringing that information from the lab to the bedside, right? How are we going to treat our patients and our families? All right, so when people ask me what I do, I often say that I'm a rare disease doctor because a medical geneticist specializes in using genetic information, right? Things like DNA tests, biochemical evaluations to help diagnose, manage, treat, or even prevent inherited genetic disorders. As a specialist, I'm trained to evaluate individuals of all ages with all kinds of different conditions. And again, because we're the only specialty that's recognized that evolved from a basic science, it lends itself really well to research and academic pursuits. So that's why we're so happy to jam together, myself, Dr. Brown, and Keolu. Medical geneticists, again, we specialize in that medicine that involves the interaction between genes and health. While some medical geneticists get really specific in terms of what they're able to study, for example, some people who are eye doctors that also have pursued genetics only focus on genetic diseases of the eye. But others, like myself in my role as the geneticist for Hawaiʻi Pacific Health, I actually care for, again, patients of all ages with a broad variety of underlying genetic conditions. We have our prenatal section that's kind of handled at the hospital through our fetal diagnostic center. But in our office, we also see cancer genetics, we see neurogenetics. A bulk of my patients are pediatric, but we also take care of our adults in the population as well. And then we have children and adults who have inborn errors of metabolism. So that falls into this biochemical category. So just touching upon from before, I know there's going to be links later on to kind of watch our previous talks, but for those of you tuning in for the first time, the big picture of DNA, right? So our bodies are made up of cells. Our cells have all of the instructions available in its nucleus to kind of tell us how to grow and develop. They're packaged into these little cute things called chromosomes. And when you unwind that chromosome, that's when you get our genetic code, right? All these letters, that special alphabet, A, C, G, and T. And those letters, again, are 3 billion coming together to tell our bodies how to grow. When we think about this cookbook analogy, again, our entire genome, so all of the information, right, all 3 billion base pairs together, that's telling us, right, what to do and how to develop. That's our cookbook, the whole cookbook, the genome. Then we talked about those packages, right, those individual chromosomes. We should have 46 in every single cell in our body. And then those chromosomes, right, have the individual genes, right? Those are like the recipes that are, again, dictating what to do. And in, you know, science and in medicine, that translates into a spelling code that makes a specific protein that will then go on to do some important function in the body. So I wanted to take a moment to touch upon family history because this is something that I do in my everyday life. When I see a patient, I think it's really critical to do so because I think not taking a good family history is bad medicine. For me, as a geneticist, right, I think it's a very powerful tool. As somebody who grew up, you know, as part Native Hawaiian and also learning the culture, it just seemed natural, right? As we think about, you know, learning about evolution and the kumulipo, and then we also kind of have to learn our ancestry, where we came from, our mo'oku oho, all of that kind of rolls into my work really nicely because I like to ask about a patient, but just beyond that, I also ask about what's going on in the family because sometimes, right, knowing what's going on can clarify a pattern of inheritance. Maybe we can see some patterns that help narrow down a diagnosis. And again, these days when I'm doing a family history, it's kind of like the first step to personalized genetic medicine, right? We're using knowledge nowadays of genetic variants that are now, right, relevant to maintaining health or preventing or treating disease, which we'll talk about a little more with respect to Dr. Brown and the cardiovascular stuff. So when I'm making, right, our family tree, we call that a pedigree, and then we use these different symbols to then kind of build a picture. And for me, identifying patterns helps narrow down what categories of disease we're talking about. Sometimes we have dominant patterns, right? Things that are, you know, passed on from every generation, every member of the family, you know, kind of can have the potential to pass it down to the next generation. We also have different patterns of inheritance. This is a recessive one, for example, and we're skipping generations, right? So again, looking outside of just the patient and again, looking many generations back helps me decide where to go next in terms of diagnosis and hopefully treatment. And then there's another type of pattern called mitochondrial, just as an example, and Dr. Brown actually recently managed a patient who had a mitochondrial disease, and this is something that we actually inherit only from our mother, right? So because of that, you know, all offspring of an infected female are going to be at some degree, you know, affected. And then we also have males that again cannot pass on the condition because again, they don't pass on mitochondrial DNA. So this is a typical pedigree for like a family that comes in to see me. And here in Hawaii, I just use this to kind of show you, you know, we sketched out what the structure, the bones of the family looks like. But also I want you to focus on the ancestry, right? We have Japanese, Hawaiian, Portuguese, Irish on one side. We've got, you know, a whole slew of things on the other side, right? Maternal versus paternal side. And this is kind of at the juncture of where I practice every day in Hawaii, right? Everyone is mixed. And I think that's really the highlight here about living in this unique, diverse place we call home. When we think about moving forward in genetics and genomics, right, part of it is how are we assessing the data? And of course, this year's, well, actually that was last year's 2020 census. This is the first time that people of Pacific Islander and Native Hawaiian background were able to actually have their own checkbox to kind of subcategorize. Before there was like a general NHPI box that you would check and then you'd have to like handwrite in what you are. But this year you could choose between, you know, Native Hawaiian, Samoan, Chamorro, and then of course other Pacific Islander as well. So within that other Pacific Islander group, we have people who are, you know, again, Tongan, Fijian, Marshallese, Palauan, you know, Chukis, Pohnpei, and Tahitian. So we can certainly do a better job of kind of doing some data disaggregation. But again, I just want you to kind of get a lay of the land. So this is data based on, again, the most recent census, and that's for the United States at large. So majority is Caucasian, and then we have a very small fraction of 1%, right? 0.02%. So like a fifth of 1% people identifying as Native Hawaiian or Pacific Islander. We also have some people that might be under that mixed races category. So again, we have a lot of admixture, so we could be captured there as a Hawaiian community. And then Hawaii, when you're contrasting that, our data is very, very different, right? Clearly, plenty people of Asian ancestry, but look at our Native Hawaiian Pacific Islander, right? 10% plus, we have a lot of admixture, right? Only what 22.4% of people are kind of mixed plate. So what does that mean? What makes what unique is that we're super diverse, but it's also somewhat of a challenge when we're talking about getting yes or no answers for diagnostic testing. And so this is some data that I presented before, but basically when we're thinking about doing sequencing or genetic testing, we have 3 buckets of results that we like to plot people into. It's either positive, meaning we've tested you for something and it came back with a diagnosis. It's negative, meaning no more. And then there's this variant of uncertain significance. So that middle category, that VUS, that's something that we see a lot in our community. And again, this is just basically to summarize that we are quite diverse and that people of underrepresented minority background do not get positive hit rates as much just because we're not in our databases. So far right under that HCG tab, that's what my families look like. So we got a lot of mixed plate, a lot of people who are Asian, a lot of people who are, you know, Hawaiian or, you know, admixed, and comparing that to other databases, you can see that again, still the vast majority of people in the databases are Caucasian. So that makes it challenging when we're trying to, again, put clues together and do DNA testing to confirm a diagnosis. And instead of getting a lot of, you know, black or white, yes or no answers, we're getting all of these gray zone question marks. So let's talk about how we make diagnoses. And again, these are just things that I want you to be familiar with. I'm not going to focus a lot, but for those of you who are interested in how we kind of diagnose before we can treat, right? We have different modalities that we use. One, which is old school, is called the karyotype. And what we're basically doing is counting up the chromosomes. So that's what our chromosomes look like. Those are those 46 pairs that we talked about. And again, when you have entire chromosomes that are missing or extra, for example, trisomy 21 or Down syndrome, you'd have a third entire copy of that 21st chromosome. There are other types of technology out there. This stands for fluorescent in situ hybridization, and we're using DNA probes to look for specific areas. So this picture is showing a target for 22q11.2 deletion syndrome. So people who have, this piece of DNA that's missing have a very specific diagnosis. It's actually the most common microdeletion syndrome that we see here in the United States and across the world. We also have this technology called a chromosomal microarray. So that is something that allows us to, again, kind of make barcodes that line the backbone of every single chromosome. So we're going from the top of number 1 down to the bottom of number 1, all the way through number 2, 3, et cetera, down through that 23rd pair to look for anything that's, again, missing or extra. We're looking for genomic imbalances. And again, compared to just that broad picture karyotype, we get really more detailed when we're doing array testing, because then it tells us exactly what genes are missing. So the genes are kind of what we're trying to get at, right? What's missing, because these are the instructions that tell us how we're going to grow and develop. And if we have a very important gene, like one that has to do with a protein that's expressed in the heart, right? Is that there? Is it not? Is it misspelled? What's wrong? And how does that affect our patient? In this day and age, genetics is getting a lot of attention because we now have technology that has opened up doors. I mean, really, we're able to do things millions of times faster. And we can kind of, again, not just look at even single genes, but our entire genome, right? So looking at our cookbook, the whole cookbook at once, versus, again, looking at that individual instruction or that individual recipe to kind of target what we're trying to find. Just as a reminder, we have these different buckets of classification, and that variance of uncertain significance in the middle, that's the one that's become, again, pretty challenging for people of minority background. Again, we're not in the databases, so oftentimes when we're doing testing, we don't get clear answers. Not every test is good for everything, and so just keeping that in mind, no test is perfect. Again, when we're thinking about our population, uncertain findings, they may be uninformative or are sometimes misclassified, and that makes it difficult for us clinically to help our patients. So let's talk about some of the ways that we can, again, bring genomics to the bedside in terms of diagnosing and then managing and taking care of patients. So here's an example. I recently saw somebody on Wednesday. This is a 26-year-old patient of mine who was diagnosed with this condition called Phenylketonuria, or PKU, at birth. Through the newborn screening, every baby, at least in the state of Hawaii and actually in the United States, should get a newborn screen. The conditions vary from state to state, but it's that heel prick test. So every baby, before they go home from the hospital, should have this card filled out, and it's screening for, again, several dozen genetic diseases or conditions. For this particular patient, she was diagnosed with, again, PKU at birth, but she's coming back to me after not seeing me for a couple of years because she's getting married soon and she wants to kind of get back on track. So beyond looking at the biochemical data, right, I can look at her blood and see what her Phe levels are, and they were 25, which is way too high, but also to interrogating that DNA to figure out what the genetic sequence difference is a part of my job. So when you have PKU, that means you have this enzyme, PAH, it stands for phenylalanine hydroxylase. But anyway, what it is that if that enzyme's not working, you cannot break down phenylalanine to tyrosine. So when we're eating, right, we eat fat, we eat sugars, we eat carbs, we eat protein. When protein gets broken down into individual amino acids, again, the phenylalanine is one of them, and we need to make tyrosine to help with other things in our body, like melanin production of catecholamines, protein synthesis, etc. And so when you have a blockage here, it's a problem. When you have the enzyme that's missing, you cannot do normal work. And so it's actually quite toxic to neurons when we don't have the Phe being broken down. Too much Phe, again, causes brain damage, and that leads to seizures, severe intellectual disability, and a lot of issues for these patients. Thankfully, with newborn screening, we're able to identify that early, and we can institute treatment, which is removing phenylalanine from the diet. It's really hard, especially here in Hawaii, right? Things like soy, any type of protein, meat, fish, dairy, eggs, all of it becomes kind of a challenge when we give it to our patients because they can't break it down, so it makes them sick. But new therapies have become available. There are pills and powders that we can use and give the patients to take right before a meal so that when they eat their, tofu sandwich or whatever, right, we are then blocking the absorption of Phe. We have this other medicine called Kuvan. It's like a vitamin that helps push that cycle along. So if you have a little bit of enzyme reactivity left that allows you to increase your protein tolerance. And again, PEG-PAL or Palynziq, this is an injectable form of the enzyme that's missing, right? So if your body doesn't make it, it's broken, we can actually give you a shot to replace what you're missing. And then that allows you to live a pretty typical life. Another example is cystic fibrosis. So a lot of you have probably heard of this condition. It's not really common here in Hawaii, but we do, see it in people of all ancestral groups. It is by far and away most common in people of Caucasian ancestry, but we see it, all over the place too. And so here's a kiddo who came to us who transferred care. She's new to the islands, and I did molecular studies to confirm that she had these specific changes here in red. Why is that important? Well, we know that this gene, right, the Cftr gene, is the instruction that makes this very important channel that helps really cells move ions across its membrane, right? So cells in the lung or in the bronchus, cells in the pancreas, right? When you have things that are trying to get in and out, we're basically having defective ability to kind of water down your mucus. So what happens is if you have cystic fibrosis, broken enzyme, you're not breaking down that mucus with liquid and it becomes kind of like concrete. That increases binding of bacteria to the cells in your lungs, for example, and then you get sick. So treatment has always been kind of attacking infection, trying to treating, the inflammation, optimizing nutrition, doing things like heart and lung transplant. But now we have new treatments. So by looking at the DNA and understanding, right, how that sequence might be broken or misspelled, we can then translate that into the cell and how that works. So on the left, you can see that there's a delta F508 mutation. So this is when you make the protein, but the protein comes out kind of crooked. And instead of folding the right way and doing its job up on the surface, because it's crooked, it gets eaten up and discarded because the body's like, oh, that one's junk. Let's not use that. So then we have medicine that can now identify what's broken. And then we're taking things back up to the surface to help facilitate better intracellular trafficking. So now we also have other things that look at the potentiators. So these are things that are, again, based on the DNA sequence and literally designer drugs based on what your DNA defect is to then help modify what's going on a cellular level to cure or treat disease. A little bit about Duchenne muscular dystrophy. So this is a condition where you have progressive muscle weakness. And this child came to me at 6 years of age. He was previously healthy, but on his most recent well-child checkup, he could not walk upstairs, jump, or stand up from a sitting position without getting lots of help, like pulling himself up from the couch, using a chair. And that's, pretty unusual. When I saw him in the office, his pediatrician had already checked an enzyme level, which is the CK, which kind of leaks from the muscle when it's having issues. And that's, 14,000 is just way too high. A normal level would be like 300 or less. And so that kind of clued us in to what his condition was. When I saw him, again, he had a lot of muscle weakness that was suggestive of Duchenne muscular dystrophy. So we did the gene testing and it came back positive. This, again, is progressive, and unfortunately, it's a fatal and degenerative disease that's caused by a deficiency of this thing called dystrophin, right? So the DMD gene codes for this protein called dystrophin, and the dystrophin is really important in keeping muscle integrity going. So heart, skeletal muscle, it all becomes really weak over time. And then, in the early phase, when kids are young, you may not notice, but by the time they're 5 or 6, they may have, some weakness, start needing help walking around. And by the time they're adult age, you have so much decline in your muscle function and in the heart and in the breathing muscles that it just becomes difficult, and life-threatening heart and lung conditions usually are the reasons why these patients pass away. Our treatment has always been kind of, using the medicines that we've had, right? Looking at steroids to help improve muscle strength, using these drugs called ACE inhibitors or beta blockers, right? To help with the heart, kind of decreasing the load on the heart so that the heart doesn't get as sick as fast. If you come in with congestive heart failure, we know how to give, things to pull the fluid off the body, give oxygen. Transplant is sometimes an indication, but, it's really not a cure for, the disease So now we have these new therapies, and it's pretty incredible to think about, right? We have this exon skipping therapy. So what that means is that we're talking down at the genetic level. We're looking at the code. On the left, you can see that the 50 is missing. And what happens is we don't have a smooth connection between 49 and 51. You can see that triangle, it's a puka. And when we're trying to read through that instruction and make that protein, it gets truncated or it comes short. So it makes some, but it's not really functional because it's cut short. Now we have this exon skipping approach where we're giving an injectable, and that medicine restores the messenger RNA reading frame. So the intent is that we know the 50 is missing because that's like the genetic defect. We end up using, right, that medicine to skip over 51 so we can make smooth sailing between 49 and 52. And so you can see, right, no pukas, no gaps. What we're able to do is kind of read through and still make the protein. It's a little bit shorter, but still pretty functional in the body. And that's what we're, again, now looking at, right? Looking at the DNA, figuring out what's wrong, and then figure out ways to kind of work through that so that the patients still have somewhat functional protein, and it really does improve lifespan. The last example that I'm going to talk about in terms of bringing genomics to the bedside in monogenic diseases today is called spinal muscular atrophy. And this condition, it's a tough one. You know, I saw this baby in the pediatric ICU, and he had come off the airplane and was really sick. He had progressive difficulty feeding, he was choking, and he ended up coming in respiratory failure because he was just so weak. When I saw him, the weakness was so profound that it made me think about testing for this genetic disease called SMA. It's also called Wernicke-Hoffmann disease. It's probably, you know, one of the more common neuromuscular diseases. And again, you have wasting and weakness due to loss of motor neurons. So these specialized cells that help, right, you kind of move. And this baby was very, very floppy. Mostly for these kids, right, supportive treatment because there was no cure. So we would do things like put in a G tube or recommend, you know, a breathing tube if that's what families wanted to kind of support the child's life. Things like, you know, surgery for scoliosis or dislocated hips, because again, yes, lots of muscle and motor neuron weakness. So we would have to do a lot of surgeries. And again, these are just ways to kind of treat the condition, but not really improve quality of life. Now there are new therapies. And again, these are all due to advances in genomic medicine. Spinraza was, you know, a really, really amazing thing that came, on the scene in around 2016, but I'm going to actually talk today mostly about Zolgensma. So that was approved in 2019, and what it is a gene therapy, right? So how is this different? Like before, I've talked about diet, right? And like removing foods that make you sick, or maybe using designer drugs, right, to treat the part of your DNA that we know isn't working right. But now we're actually using gene therapy. So what this is that we had a viral vector that was introduced right to the patient. It's a one-time dose, and you give it through an infusion. And what happens is these viral vectors go into the body and they insert this SMN1 protein. So in people with SMA, their SMN1 is broken, and that's not working. That's what leads to the motor neuron loss. That's what causes the phenotype. Now we're able to use gene therapy and using viral DNA containing the right instructions to make a functional SMN1, and that inserts in the body, and you're able to make some on your own. It's a one-time dose, and then you trick your body into making, right, what's broken. And it's incredible. People who've gotten this treatment with Zolgensma with SMA type 1 have not needed breathing support. 91% of individuals in this first study trial were alive and did not need like a breathing tube, or most would. 60% of kids with this very severe disease could sit up without help. And that was compared to like 0% of the kids who didn't receive this gene therapy. So it's currently approved for kids 2 years of age with a genetically confirmed diagnosis of SMA. but it's also really expensive. And when I say really expensive, I mean super, super chokis expensive. So the one-time shot, it is $2.1 million. And I think that kind of highlights, again, with advances in technology, we're able to kind of figure these things out, but now come the disparities and inequities in healthcare and how we can kind of think about that as our next step to help families get the treatments that they need. Again, just through these cases, I wanted to highlight how we bring genomics to the bedside in monogenic diseases. Again, diet, or now we're talking about specific drugs or medicines that were designed to either replace what was missing or to skip over the DNA mistakes so that you can start making your own, or even gene therapy, right? It's here, and I think there's a lot more coming down the pipes in the future, but again, changing someone's inherent DNA so that we can, again, trick your body into making what's broken on its own so that we somewhat have a treatment. You know, maybe not a cure. We're not, you know, doing gene editing for all bazillion trillion cells in your body, but we're getting somewhere. And it's, you know, I'm really hopeful for the future. So again, I specialize in the intersection that involves genes and health. I think we're now starting to reimagine the treatment of genetic diseases due to the breakthroughs that we have in genomics. And again, these were for monogenic diseases, right? Genes that have one problem that we can kind of isolate and then treat. Dr. Brown's going to talk a little bit about cardiovascular disease, again, with an increased disproportionate burden in Native Hawaiian Pacific Islanders and what that means for the future of health. So just to summarize, there's like vast amounts of data out there. I think in order for us to treat a disease, we need to be able to diagnose it. And for those of us who are of minority background or mixed ancestry, what we now must bring is equity and parity and more data, more research to the table. That's going to require more dollars, but we need to do this. We owe it to the people of the Pacific so that we can, again, ensure that the maximum potential of these benefits apply to everybody. Anyway, I'm going to stop sharing so that I can get Dr. Brown going. Give me 1 second. Thank you so much. Stop sharing.
Wow, Leah, that was fantastic.
Great. Oh my goodness. No, Stacy, thank you so much. Am I, Keolu, am I unsharing?
Yes.
Yeah, you're all good. My Ka'i, that was lovely. We have so many questions, but we're going to wait until after Dr. Brown finishes, and then we'll have more of a wala'au conversation.
Yeah, Leah, really wonderful. Thank you for that talk. I just figure, am I sharing my slides?
Yes, we can see them.
Okay, all right, good. Yeah, I am really just so honored to have been invited to partake in these sessions because they are a terrific learning experience for me. So thank you really to Leah and Keolu and JoAnn, and nice to meet everyone else here. I look forward to getting to know the Ola Hawaii community more and more. So I am going to talk about cardiovascular health disparities and the role of genomics there. You've heard already about why genetics matters when we talk about health, and that it matters when we talk about health disparities, when diseases disproportionately burden some communities more than others, communities that may be less well served by current health systems or understudied with regards to the cutting edge health technologies that we're talking about. I'm going to focus on cardiovascular disease. As Keolu mentioned earlier, the field is working to harness genomic technology to address both rare monogenic diseases that Leah just told us a lot about, as well as common complex diseases. So cardiovascular disease is super common, and from a genetic standpoint, it is a complex disease. Before I get into the weeds of it, I want to briefly introduce myself and what I do and why these topics are just becoming relevant to the health issues that I deal with as a clinician. To be clear, in this genetics webinar, I am not a geneticist. I am a medical doctor whose specialty is neurocritical care. I take care of people with sudden and severe brain injury, and I work in an intensive care unit or ICU at Queens that specializes in supporting patients who are critically ill as a result of various types of brain injury. By far, the most frequent and major cause of brain injury that I take care of is stroke. So this talk is going to focus a lot on stroke. Now, when you hear that somebody has a stroke, you may not immediately think of that as an issue that is genetically determined or hereditary, right? It comes on all of a sudden, usually later in life. But as we are rapidly learning in this age of high throughput gene sequencing technology, a substantial percentage of the variation in stroke susceptibility is heritable. And we are just beginning to explore how we can harness that degree of heritability to improve stroke prevention and treatment. So back up a little just to get everyone on the same page. What exactly is a stroke? It's when someone suddenly becomes disabled, right? They may become weak or paralyzed on one side of the body, not be able to talk, and it's an emergency because when we can get to the hospital right away, ASAP, we can provide treatments and interventions that may reverse or minimize the long-term disability from stroke. The acronym we use to spread awareness and teach people about stroke is FAST, FAST, and it stands for face weakness, arm weakness, slurred speech, and time, because time is of the essence. When a stroke is happening, you need to call 911 right away to get the best chances of a good recovery. Okay, but why does a stroke happen and why is it related to cardiovascular disease? Well, the stroke happens when a blood clot either forms or gets lodged in a blood vessel that supplies the brain. The heart pumps blood and oxygen to the brain, and the brain needs this every second of every day to survive. When a clot forms in a brain artery, that plumbing system gets clogged, the brain doesn't get the blood and oxygen it needs, and it begins to die. Because the brain controls not only our thinking, but also our body's movements and strength, when this happens, we see disability in the rest of the body. So the reasons that someone might have a stroke then have everything to do with the heart and the blood vessels, cardiovascular health. The same risk factors that cause heart attacks cause strokes. High blood pressure, diabetes, high cholesterol, obesity, smoking, drug use, all of these diseases are the real problem when we talk about stroke. So we mentioned that cardiovascular disease is common. It's also deadly. This graph represents statistics from the American Heart Association. It portrays the relative frequency of major causes of death. Cardiovascular disease is here on the left. It is the tallest bar. So as you can see, it kills the most, more than cancer, which is second. In Hawaii alone, it is the cause of roughly 4,000 deaths per year. So if we're going to talk about saving lives and saving as many lives as possible, this is the disease to work on. But it's more than just curing disease left and right, though, because cardiovascular disease is not fair. It does discriminate. It kills and affects people unequally, and in every region of the world, there is disparity between groups of people and the burden of disease that they deal with. So some people have the most resources to help them prevent, treat, and recover from disease. For others, those same basic resources are out of reach. In Hawaii, when we look at our cardiovascular mortality and try to pinpoint who in our state is most at risk, most vulnerable, we find something alarming. People who identify as Native Hawaiian have the highest mortality rates and the highest rates of contributing diseases like diabetes and high blood pressure, and they become disabled from stroke at much younger ages. There are other groups here that also suffer a high burden of disease, Filipinos, Micronesians, but the point is that it is not equal across Hawaii residents. Many of the reasons for these disparities fall into the categories of so-called social determinants of health and have to do with economic resources, access to appropriate healthcare, health education and awareness, culturally relevant lifestyles and diets. There are a lot of potential ways to look at it that are all super important. However, I'm also here to tell you that some of the risk is inherited, that genes and the particular variants we may be born with have some influence on the baseline level of risk that we each face, which can then be further modified during the lifetime by different environmental exposures. Let's focus for a second on the cardiovascular disease that I deal most with, stroke. When I am caring for someone with stroke, part of my job is to help them heal. And another huge part of my job is figuring out why that person had their stroke so that I can help them prevent another one from happening in the future. We already know about several common stroke subtypes, which may be mitigated by different chronic diseases. For example, high cholesterol and buildup of plaque in the arteries, high blood pressure, atrial fibrillation or an irregular heart rhythm, and a predisposition to forming clots in the veins and the arteries. All of these things can contribute to stroke risk. We have seen that the kinds and causes of stroke that clinicians already know about each have associated genetic loci. We know this. And some of these loci have effects that overlap with other stroke subtypes. Notice that in this diagram, we're highlighting 32 genetic loci. It's not one gene that causes stroke, but many genes that contribute to the risk of stroke. So Leah just taught us all about important monogenic diseases, diseases that are caused by a mutation in one important gene, which then leads to the clinical expression of disease. These diseases follow the rules of Mendelian Genetics, which Keolu mentioned, so named after Gregor Mendel, who in the 1860s experimented with pea plants and found that certain characteristics like pea pod shape and color would skip generations and turn up more often one way than another. He established that certain traits can be dominant or recessive. And the diseases that Leah talked about can similarly turn up in dominant or recessive patterns within families. As we've been alluding to, common cardiovascular disease is not like this. There is not one gene that is responsible for whether someone has a disease or not. But a person's risk of developing disease relative to another person's can be explained to some degree by genetic variation across many different loci. We call this kind of heritability polygenic, many genes. Genetic risk is driven by millions of genetic variation scattered throughout the DNA, and these are each called single nucleotide polymorphisms, or SNPs for short. Each SNP only contributes a minute amount to disease risk, but when added up, the small changes can have a big effect. So for diseases like this, we are not looking for a single variant that will cause specific disease in 1 to 2% of the population. We are looking for thousands of variants that may be prevalent in 5 to 50% of the human population, which pulled together mitigate risk of very common diseases. We can pull these together, the SNPs, that we know that we pull together the SNPs that we know contribute to disease risk into something called a polygenic score. Something the field is working on is perfecting polygenic scores and the way we use them so that ultimately a person's overall genetic risk estimate in combination with other more traditional medical data like history, blood laboratory levels, and imaging combined together can help us to understand an individual's own unique level of risk for having disease and the urgency of implementing strategies to reduce that risk and prevent major events like strokes and heart attacks. This figure demonstrates the performance of a polygenic score for coronary artery disease, which is when the arteries feeding the heart become diseased and harden and at high risk for a heart attack. You can see that above the 70th percentile of this polygenic score, which is on the horizontal axis, there begins to be a steep increase in the risk of coronary artery disease. So the high score predicts the disease. Similar curves have been demonstrated for atrial fibrillation or irregular heart rhythm, type 2 diabetes, inflammatory bowel disease, breast cancer. Again, the high score predicts the disease. My friend and colleague, Juliano Costa, looked at whether polygenic risk score for major cardiovascular events like stroke and heart attack could predict risk specifically in people without known traditional risk factors. So middle-aged, healthy adults in their 40s to 60s without known diabetes, high blood pressure, high cholesterol, or the like. The different colored lines here represent the different genetic risk categories. So blue is high risk, green is intermediate risk, and red is low risk. Time is on the horizontal axis, and strokes and heart attacks are the events on the vertical axis. So what we are seeing here is the separation of the lines as time goes on. As people get older, the risk of having a stroke or heart attack is significantly different depending on their genetic risk. Now, although a lot of more work needs to be done to make this kind of tool useful in a doctor's office setting, you can imagine how, especially for those folks who don't have traditional risk factors that they know about, a way to predict how high risk they are for having a stroke or heart attack can help people and their healthcare providers focus their prevention strategies where and when it really matters. In addition to prevention, polygenic scores may play a role in treatment, so tailoring therapies to individuals based on their own predicted responses to different therapeutic options. Take, for example, a stroke survivor, so a person who suffered a stroke but made it through and they're looking forward. We know they already had a stroke and they definitely are at risk for having stroke. They could have another one, right? So the goal is to prevent that. The association between stroke and high blood pressure is well known, and keeping the blood pressure in a normal range is crucial to reducing the risk of another stroke. In this study, my colleagues found that high genetic risk, dark blue on the right, versus the low genetic risk on the left, associates with the likelihood of having resistant hypertension or blood pressure that remains high in spite of using multiple blood pressure medications. These blood pressure medications I speak of exist in many different classes, meaning different meds act on the body's blood pressure through different mechanisms. Our strategies for managing blood pressure are not one size fits all. One person may do very well with a certain medication, but that same medication may not work at all for a different person. So genetics may help us tailor therapies to individuals. Studies I'm highlighting here illustrate how we can use genetics as so-called experiments of nature. If a certain drug leads to lower blood pressure through, say, blockade of calcium channels, we can use a mutation that results in calcium channel inhibition to simulate the effect of this drug class on preventing strokes. We can test different types of medications this way and discover which medications achieve the goal better. When someone does not respond as expected to a certain medication, it's called a drug therapy failure. So why that happens in some people may have to do with genetics in certain circumstances. Let's take, as an example, a very popular medication for diabetes, metformin. Metformin is prescribed as a first-line treatment for diabetes to almost everyone with diabetes and normal kidneys. It is a great medication and helps a lot of people, but not everyone. Diabetes genetics researchers have found that the response to metformin is 30 to 40% heritable, and it started with this Genome-Wide association study. What I'm showing you here is a Manhattan plot. So you see how it looks like a city skyline with the dots reaching to different heights on the graph. The vertical distance represents the statistical significance of a genetic locus's association with the trait of interest. In this case, it's the response to metformin, and then the different genetic loci are spread out along the horizontal axis. So where the dots land above this blue line is a statistically significant locus that's been identified. Since this study, which found signal near a certain locus, the exact gene and what it does has been identified. So knowing whether someone will respond to the medication or not using genetic tools can help us make sure that patients are in fact getting the right medication to actually treat their disease. Pharmacogenomics is what we call this field of using genetic information to tailor medication strategies to an individual's likelihood to respond to that medication. Another example, Hawaii had a publicized experience with a drug called Plavix or clopidogrel, which is a common medication used to prevent clot formation in small arteries and prevent heart attacks and strokes. The way Plavix works is that it relies on liver enzymes, so the own body, to metabolize the drug into its active form. Some people have a mutation in a certain gene that reduces the function of this enzyme. So in those people, Plavix is just not going to work. Taking it in those people as a prevention medication would be a false sense of reassurance. In the general population, this mutation is rare enough that in the bigger picture, it is still worth using this medication. It's a good one. However, it is less rare in populations of Asian descent. Hawaii has a lot of Asians, and that is the background for the lawsuit. But my point being here is that, being able to test for the genetic mutation would make it possible to know for sure whether a certain patient has Plavix as an option or not. The kinds of strategies I've talked about using genetics for disease risk screening, using it for therapy selection are broadly referred to as genomic medicine or precision medicine. Innovative health systems are already applying these strategies in small increments. Here are some headlines of notable places and their programs. The point is that genomic medicine is actually here. We often talk about it in futuristic terms, but the future is here and we need to get ready for it. So far, based on what I've shared with you, it might be easy to think, well, great, we're advancing technologies and making medical care better and more precise. So let's do it, right? But there is a problem. And here in Hawaii, as well as our neighbors in other Pacific Islands, we have a specific version of the problem. The problem is the research inequity. So we've had now about 15 years of discovery with high throughput gene sequencing technology, and nearly 90% of all participants ever enrolled in genetic studies have been of European ancestry. You've seen these figures before in the earlier episodes. The disproportionate representation of the global population in genome studies is completely relevant to everything I've talked about so far, to how well precision medicine strategies are going to work and to how well they will work when it comes to cardiovascular disease, the #1 killer and the killer that disproportionately affects scientifically and medically underrepresented populations like the people of Hawaii. Those polygenic scores we discussed, their performance and accuracy is low when used in target populations that are of different ancestral descent from the populations that these score components were discovered in. This figure is from a study that compared polygenic score accuracy within and between different populations in the African continent. Derived from studies comprised mostly of Europeans, these scores do not apply as well, and they apply to varying degrees in non-European populations. So in the coming age of genomic medicine, what are we most afraid of? That the social determinants of health inequalities will soon also be compounded by inequality in the availability of accuracy of precision medicine strategies. So I'm taking the slide straight from Leah's talk and reiterating, both for monogenic disease screening and knowledge derived from population genetics, we need to prioritize inclusive research in order to avoid exacerbating health disparities. And this is true everywhere, but uniquely so right here at home in Hawaii because of the truly unique ancestral makeup of many of our residents. The task ahead is no small feat. However, to scan 40 million variants across the human genome and detect single SNPs that have even the slightest association with disease that matters only when expressed in combination with thousands of other variants. These are not studies we can do in a couple hundred people and be done. across the world, the challenge of sample size for these genome-wide association studies is well known. And in the last decade, scientists have changed the way that they do this kind of science. Science is no longer about running a secret experiment in your lab or basement out of fear that someone else is going to steal your ideas and get all the credit and reward. Science like this has to, by definition, be collaborative. Genomic discoveries physically, statistically, cannot be made by one researcher or one country for that matter. So for different diseases, people have joined together and formed consortia for the common goal of advancing knowledge. And for the discoveries I mentioned, not one person has received credit. It's been groups of people from all over the world working together. I am, for example, a member of the International Stroke Genetics Consortium, and it welcomes anyone else with interest, something to learn, and something to contribute. These types of consortia, international consortia, facilitated the discovery of brand new genetic risk loci, because when we include people of multiple different ancestry in these studies, we are no longer restricted to alleles that are only frequent in European lineages. We may find variants that happen to be more frequent in groups of people from one region, and those variants are going to be important for those groups of people, and they have to be discovered, because discovering new genetic loci leads to the discovery of new mechanisms of disease. When we know that a certain gene can increase disease risk, we can look at what that gene encodes and the biological processes that it mediates. Every step of the biological process discovered may become a new drug target, a new understanding of the cause of a complex disease, and why certain groups of people may suffer from it more than others, and how we may begin to fix that. Are any of you wondering at this point, what might be discovered if we were to look for genetic variants associated with cardiovascular disease in a group of Hawaiians? I did, I wondered that. I knew that we don't have the sample sizes. The number of Hawaiians who have participated in genetic studies is very low, and there are good reasons for that, reasons that make it an important challenge to advance the field of Hawaiian genomics. We talked about these challenges in the earlier webinars, and we must continue to do so as we creatively explore homegrown solutions to independent, sustainable ways to own and advance Hawaiian genomics on Hawaiian terms. So those of you listening and engaged with this webinar series are going to be the leaders in this effort. That was a tangent anyway. A minimal amount of genetic studies that have included Hawaiian participants does exist, and I used it to perform a genome-wide association study. So remember that Manhattan plot I showed you earlier? This is another one generated by my own analysis. Again, so all the genetic loci spread out across the horizontal axis and the statistical significance of the association on the vertical axis. And so what pops out here is one genetic locus that has not yet previously been described to be associated with stroke and heart attack. Am I claiming that we have discovered a new stroke locus? No, there's not enough data to have confidence in this finding, but also sort of yes, what we are claiming is that research inclusive of Hawaiian genetics is feasible, likely to be fruitful, and potentially very important. This locus encodes a histolysine N-methyltransferase, which has transcriptional regulation and oncoprotein or cancer-related functions. Variants discovered in studies that are inclusive of non-European populations will be important for improving the accuracy of our genetic tools and applicability to underrepresented populations. In addition, they might also be important for humankind in general. Multi-ancestral analyses advance knowledge relevant to the populations included, and they also advance knowledge relevant to everyone, because ultimately across the globe, we are talking about humans, all humans, who are 99.8% genetically identical. So we need to move forward for good reasons, and we need to move forward carefully, balancing and towing the lines between global contribution, collaboration, and data sovereignty. You are informed from participating in this webinar of important historical and ethical challenges that Native peoples and island populations face in particular when it comes to genetic data sovereignty, determination of research priorities, and ownership of the discovery process from the ground up. This is an incredibly complex challenge, and people like Keolu have amazing ideas, but whatever way we look at it, it's going to require initiative coming from within the communities who happen to be most affected by the current inequities. Seems hard, I know, but the only impossible journey is the one you never begin. So here we are beginning, and I look forward to seeing what our communities will accomplish here in Hawaii. So with this thought, I will conclude. Mahalo nui.
Awesome. Crystal clear. Both amazing talks. Very, very edifying. I hope everybody enjoyed this opportunity. Just one, for education, but two, it's the way you guys put it together really makes sense. So smooth. And I guess we'll begin with questions. I personally have a bunch of questions, but more comments. And I'll go with our first question here from the Facebook Q&A, I'm guessing, and it's from an anonymous attendee. And they said, when a person has a stroke and is admitted to the ER, what type of medication can help treat the stroke?
Great question. Great question. So the key, as I mentioned before, is time, right? The quicker that we can get to a stroke, the more therapies are available. So in the situations where we know exactly when that stroke started and we got to it but within 4.5 hours, we do have a therapy called IVTPA, which is available. And that is a medication that's infused. Remember how I described stroke as a blood clot in the arteries of the brain? So the goal of the medication is to bust that clot. It's a clot busting medication. It does have some risks because it busts clot. It can cause bleeding. But in certain patients, the risks and benefit balance weigh in favor of using this medication. And when we do give it, patients need to be monitored in the intensive care unit for all those potential problems, but majority of patients do have better outcomes when TPA is used appropriately. So that's one. And another that is only available at Queen's is something called thrombectomy. And it's actually a new intervention that has become the standard of care for large strokes in particular, just in the last 6 years or so. But it's a procedure where we actually physically use a catheter tool to go thread through the arteries, starting from either the wrist or the groin, and it goes up to the head and it physically pulls that clot out and opens up the blood vessel, allows the blood to return to the brain. Again, very specific populations of patients will benefit from this procedure, but again, as long as we can get to patients quickly, we have very dedicated imaging modalities to try and decide where that risk-benefit balance falls. And for patients who are eligible, it can be life-saving. So those are our two main acute therapies for stroke.
Wow, I always actually wondered about that on my end, but that's a lovely, elegant answer. We'll give another question here we have for Dr. Dowsett, and it says, mahalo, Dr. Dowsett. We're interested to learn more about Prader-Willi syndrome. Any idea about the stats in Hawaii, any cases, frequency, etc.? And I think that's a really interesting question. I have no idea. Do you know?
Yeah, sure. Great question. So this is one of those things that, we see here in my clinic. So I have a handful of patients, I would say fewer than 10, that vary across many ages, some, younger than others, closer to the adolescent group. And I think we also care for one adult, but this condition, the Prader-Willi syndrome, is basically a difference in the DNA that has to do with imprinting or the way that different genes are turned on and off. And so on chromosome number 15, there is a very, very specific area. And then just depending on whether you express that information from mom or from dad, that leads to different conditions. So when we talk about balance, right, we talk about inheriting one set of things from mom and one set of things from dad. And when you have two moms on versus two dads on, you can get, again, syndrome. So when you have Prader-Willi syndrome, you have an imbalance on chromosome 15. You're missing the information that dad's supposed to be giving you, so you express mom twice. And so what does that mean? These children often present in the NICU with low floppy tone. They have poor feeding. They're pretty weak. And will often need interventions like a tube through the nose to feed or a G tube. And then the inverse happens over time as these kids get older to like 2 or 3, they end up having this insatiable appetite. That's, again, what we call hyperphagia. Children cannot stop eating and they oftentimes can become obese. Now, in terms of the management of that, right, typically we would initiate growth hormone therapy. It's been used in the treatment of Prader-Willi syndrome for a long time. And what it does is with growth hormone, right, you're doing a lot of things, but you're also increasing your lean body mass. You're, again, cranking up your body's metabolism and ability to kind of, again, burn up calories. And so by giving growth hormone, you're kind of, again, replacing what's deficient, because that syndrome, right, the dad part's not expressed and then you don't have the growth hormone going on as normal. So for those of you who aren't familiar with PWS, that's what it is. And in terms of looking forward, in terms of research, great question. So now that we have ability to diagnose this, before it was all clinical diagnosis, right? You have a floppy baby and then you kind of watch them over time and then they start to eat plenty. That's how you give them that clinical diagnosis. Now we have ways to look at the DNA, and there's actually many different ways you can get Prader-Willi. You can have a microdeletion on chromosome 15. You can have turn on, turn off, right? Or methylation imbalances. You can have all kinds of stuff, uniparental acetyldiestamine, but anyway, I digress. Once we identify what it is, then we can treat. Not only is growth hormone important, right? So we want to get the baby to see an endocrine doctor as soon as possible, but getting different types of interventions as well. There are many clinical trials that are ongoing actively. Most are on the continent. So places like California and New York and many sites, kind of across the continent where you can enroll in different phase clinical trials. So phase 2, 3, and 4 for different types of, again, pharmacologics or medicines that you can take that will help address the things like the hyperphagia or the eating too much, things like the obstructive sleep apnea and some of the differences that kind of go along with that beyond just putting on a breathing mask. They're also looking at different types of medicines, like even cough syrup, like a guanfacine, to try and see if that helps with some of the behavioral undertones that go along with the disease. So I think kind of A, knowing how to diagnose, and then B, using that information to then see what's available for treatment is important. I will say a lot of our families, are here in Hawaii. And so if that's something that you're looking for, you're more than welcome to reach out or message me. Again, I manage patients with this all the time. And we have a small network of families who beyond, the Facebook and beyond the internet support groups and beyond the national foundations, it's nice to connect with somebody kind of in your big backyard. So we can kind of help facilitate that through Zoom, but also kind of, again, thinking towards the future of being able to pick this up sooner so that we can kind of, again, treat as early as possible using that precision medicine piece like Dr. Brown was talking about. I think that's hopefully where we're headed for PWS, Prader-Willi.
Thank you for the comprehensive response. I really appreciate that. We have a ton of questions now, so it's just flooding in, typical Hawaii. So here we go. The next one will transition back to talking about cardiovascular disease. And I don't know if this one is from Cliff Kopono Jr. or Senior. Is it from Dr. Cliff Kopono, the top 10 surfers in the world? Is he with us here on Saturday? I don't know. Maybe it's his dad, but he says, given the small population of Hawaiian gene pool studies relative to CVD, which are negatively high, what prognosis would you address diet as a preventative modality for DVT, for example? Great question. And Stacy, take it away. Okay, it's Cliff's dad, Raja, the senior. Right on.
I just want to make sure I understand the question correctly. So what prognosis would address diet as a preventive modality for DVT? I think there's a lot of ways to connect all these different concepts, but the major point being that diet, in addition to a bunch of environmental exposures, lifestyle modifications, have been shown to be able to alter the risk, alter the rate of events despite a genetic risk category that may be intermediate to high. So knowing the overall genetic risk is not a, it's not a here's your fate kind of thing, but it is informative so that things both in the medical strategy as well as the lifestyle strategy categories can adjust that overall lifetime risk. So it is still important.
I tend to agree with you. Big time, a lot of factors, a lot of variables that contribute to this. Eating healthy, sleeping healthy, you know, no stress. Yes, mahalo. All right, so we have a question from Kauka Dr. Marjorie Mao, but it is under Mariana's name here, and it says, in a perfect world, how would you folks create a cohort that could answer some of the genetic related health questions that are relevant to our populations here in Hawaii? And I'll let you, both of you guys add to this, but I think it's a timely question. And I think Dr. Mao knows what we want to do. And it's going to be a lot of fun. It's going to be a very exciting future, but I'll let you guys explain what you think would be an ideal situation or scenario for expanding a project like this.
I mean, I completely agree with what Stacy or Dr. Brown had said earlier about collaboration, right? That's going to be really important, but also, you know, getting the community buy-in is a huge thing as well, right? We want to have meaningful impact on the community, but in order to do that, we have to make sure that we have you guys engaged from the beginning. I think one thing that I do want to mention is that in order to do that, it does require dollars, right? Research dollars, things like that. And we certainly want to make sure that, like Dr. Brown said, the time is now. You know, we are in the genomics era and we do not want to be left behind. So there are things I think that we can do to help think about bettering the future for people of the Pacific, including a genomics institute that could hopefully, again, bring this data together with keeping all of these various factors in mind. And again, we want to do good science that we could bring, you know, great answers back to the best people in the world.
Yeah, that's everything that I am thinking of too. And I'll just add that as we build such a genomics institute, it's also important that we're training up, right? And so we're obviously going to need collaborators, we're going to need guidance, and we're going to also want to hire, but we want to do that all within the infrastructure of who is determining the priorities of this research. And so that kind of capacity building is going to be super important to make this venture a lasting, sustainable program.
I 100% agree. I think from the infrastructure point of view, we really have to focus on creating and vertically integrating all of the technologies and personnel. So when we outsource things to USC, for example, that doesn't help us because maybe there's a handful of Hawaiian people who live there, but really we need to be in control of all of the systems and the data. And a lot of our partnerships and kind of colleagues are in search of authorization and not consensus building within our lahui. And that's exactly what we're doing here. And I think it's really cool to have everyone else's input. Obviously, we have these seminars so that people can provide us with suggestions and ideas and things that we can avoid so we can avoid failure. So yeah, that's my thoughts. Okay, we got another one here, and it says, Dr. Dowsett, can you talk about coffin Cirrus syndrome? Never heard of it. How many people in Hawaii have it? Which gene is affected? Do you, Hawaii Genetics Clinic, do you follow up with adults who are diagnosed with coffin Cirrus syndrome by the clinic? when they were younger, etc. And then I have a follow-up question to that, which is do we know of any Mendelian disorders that are specific to the Hawaii Islands? And can we anticipate the discovery potentially of new ones in the future?
Great questions. Thank you so much, Keolu, for kind of adding on to that, because I think we'll cover all of it. So the first part for Coffin-Siris syndrome. So that's a condition that, again, single gene, but actually somewhat polygenic in terms of it's actually not just one gene that we know causes the syndrome. There are many different genes, right? So there's separate instructions, but they kind of work together in a network. So when you have any one of these genes in the network that are wrong, whether it's one of the SMARC genes or ARID1B, for example, these all, again, when you have a spelling mistake or a piece that's missing or whatever it is, when the protein cannot get made and it doesn't interact in this network of other proteins, that causes Coffin-Siris. So things that we see are, multisystemic, meaning the gene or protein is often expressed in many different tissues in the body. So these children often have, difficulty with tone. Sometimes we need help with feeding. We'll also have, some learning differences. Sometimes we have to think about, the way the heart is structured, or is the gut normally, right? Like going from tip to tail in the normal fashion, or is that twisted about in a condition called malrotation? Some of the features that we see on the outside include, a difference in eye shape and a difference in nose shape. Because of the low tone, you have like a downturned mouth. You might have very, very hypoplastic pinky nails, things that, again, on the outside, somebody with dysmorphology training can kind of pick out and then select the right molecular test to get a diagnosis. Again, there are a lot of tests out there, but I think, instead of testing all 20,000 things at once, right? Hopefully we're looking at each patient individually and kind of tailoring what we're supposed to do to get the highest yield for answers. How many patients would I say? I mean, now that we're having more availability of genetic testing, I would probably, we have fewer than 5 patients with Coffin-Siris. And yes, I see patients of all ages. So, some I diagnose baby time, just because their medical complexities keep them, in the neonatal ICU for ICU level care. And that's how we pick it up. Sometimes, these kids are coming to see me at several years of age, and they've kind of got a lot of clues that kind of help with a diagnosis after a long diagnostic odyssey. But, I follow adults too. So, so whoever is out there asking about Coffin-Siris, yes, by all means, reach out. If you go to the Hawaii Pacific Health website and you can look up, myself or Hawaii Community Genetics, we're more than happy to accept referrals for patients of any type of genetic or Mendelian disorder, including Coffin-Siris. So if you're interested, by all means, yeah, we're more than happy to help out. And then, somebody I trained with, she works in Virginia. She's a geneticist there. Her name is Dr. Samantha Vergano, and she actually runs the data registry for Coffin-Siris. So even though we're talking about a super, super rare disease where you only have a few hundred cases ever described, we have somebody that has that expertise of seeing the mild and the severe cases and trying to putting it all together. So anyway, so reach out, let me know, and I'm happy to kind of help in any way that I can. So Keolu's question, right? Is there anything, and I get this question all the time, when we talk about Hawaii and specifically like Native Hawaiians, or, asking questions about like people who are living on Ni'ihau, and is there like this rare thing that we see in the Hawaiian population? And I would say, no, there's no specific disease that is unique to Hawaiians. We know that, for example, we talked about cystic fibrosis, right? And we know that's common in people of Caucasian ancestry, but it makes sense because, what happened was back in the day, right, the Norwegians and the Vikings who are coming right from the north and doing spice trading through India and the like, they would be carriers of cystic fibrosis. So instead of actually manifesting the disease, one copy of their chromosome was healthy, but the other one had this inability to function appropriately. And what that causes, right, so you're not sick with cystic fibrosis, you're a carrier. But because it was a channel issue, right? We're talking about chloride channels. When you got sick on these long trips, trading spices with India, and you got dysentery, right? Or cholera, you were at an advantage as a carrier because then you wouldn't die from having all this explosive diarrhea, basically. So what that boils down to is that it became advantageous for certain populations, right? To kind of carry that trait. And so same reason why we think about malaria in parts of Africa or the Mediterranean, right? Reasons why we have this, what we call heterozygote advantage. It exists all the time in nature. Here in Hawaii, we haven't identified anything that is specific to whether it's Native Hawaiians or even greater Polynesians or even people of the Pacific. However, I also completely think that Keolu could probably help answer this too, but there are so many things that we don't even know. I mean, recently there were some papers about interrogating the genome of people of, different indigenous background. We're finding genes that we didn't even know existed because we've never really looked at people of minority backgrounds. And so it's possible, but right now there's nothing that I would say that's like a, Native Hawaiian disease, but a lot of the things that Dr. Brown was talking about, right? These polygenic things that have to do with, diabetes, hypertension, hypercholesterolemia. There are a lot of different factors that I think play into the diseases that we know plague our people too.
Yeah, awesome response. I guess we'll switch back briefly to thinking about cardiovascular disease. And this question is very interesting. We know that there are all of these sort of dialectically intertwined relationships in common complex disease. Some people call it comorbidity. And I'm wondering, this question, it's from, it says, what is the intersection between cardiovascular disease and Parkinson's? Does one cause the other, etc.? And I'm wondering if you could kind of elaborate on that, Dr. Brown.
Sure, yeah. So several ways. One is that in addition to straight up what we call idiopathic Parkinson's disease, which is a neurodegenerative disease that causes those symptoms, there's also Parkinsonism, which mimics Parkinson's disease, but may be caused by many different things. It can be drugs, exposure to drugs, medications, and also vascular events. So cerebrovascular disease itself, meaning if there are imagine many silent strokes that don't present the way I described, like big strokes you're coming into the hospital for, but over time accumulate and lead to atrophy and brain dysfunction. Sometimes, depending on the parts of the brain that are affected, it can end up looking just like Parkinson's disease. So there's that. And then, of course, more and more we're finding that cerebrovascular disease will contribute to all sorts of cognitive decline. And so this whole picture of dementia, Alzheimer's disease, other degenerative disease, vascular dementia, in our overall aging population, right? One of our big research priorities is going to be how do we address the frailty and cognitive impairment that people develop over long amounts of time that may very well have everything to do with those vascular comorbidities. And so when we talk about using these strategies to prevent and treat stroke, we're also talking about using them to prevent silent cerebrovascular disease. Stroke is the tip of a giant iceberg that is relevant to our growing and aging population. And so I didn't even begin to get into that in this talk, but it's all related along a spectrum. And so the priorities are, they spread across both stroke, specifically heart disease that's related, coronary disease, and things like dementia and geriatrics. So in all those ways, it's related.
All right, guys, I have some questions that I've been burning to ask you now that obviously we want more questions from the Lahui, but if they don't come, here we go. Okay, one of them is, we talked about all of these new, amazing, emerging therapies, none of which seem like they're personalized or tailor-made for our community. And I'm wondering, that seems like it's very much the future. How do we, one, position ourselves in a way that we can be in vertical control of many of those technologies so that they serve our community and our people? And then two, if there are all these amazing new types of drugs, you talked about a drug that costs $2.1 million, Dr. Dowsett, that's nuts. That's against the common rule that we have basic problems where people don't even have access to healthcare. And there was a new Alzheimer's drug that just hit the market that was hardly effective. And it's going to hit the market at $57,000 a year. And another, another cystic fibrosis drug that hit the market at $300,000 a year from Vertex Pharmaceuticals. So one is, what kind of solutions can you guys suggest so that we can make drugs that are just as effective, but we can provide for our community at cost or accommodate their insurance programs? Or do you have any solutions for the future?
Oh man, good question, Keolu. I mean, I think we certainly have discussions about how we could kind of think about the future of the Lahui, but I think it's also important for us to make sure that we're getting good data. Just like Dr. Brown's example of using the clopidogrel and learning about, how people of different ancestral backgrounds, how their liver enzymes work and may not be as effective, right, in terms of tolerating or metabolizing or clearing out, a specific drug, whether it's effective or not. Those are simple ways that we can kind of institute screening. I think one of the things that I don't have a solution for, but I know is going to be a challenge in our community is that, again, we're admixed. So, when we're doing these pharmacogenomic studies, I think we have to keep in mind, where are we focusing in on the genome, right? For someone like that pedigree that I threw up, that's like Hawaiian, Japanese, Puerto Rican, Irish, Filipino, are we looking at the liver enzyme, and does that liver enzyme, live in the Caucasian part of this person's genome, or is it housed in the Filipino part of this person's genome, or is it, the Hawaiian side of this person's genome? And again, using that information, right, using that level of genomics to then, one day, A, recognize, and then B, tailor medicine. I think it's possible, but building that infrastructure, getting the data, and again, doing all that bioinformatics is going to take buy-in and time, but we can do it.
Yeah, there are smarter people than me doing those bioinformatics analysis and everything. But, their perspective is that the admixture is an advantage. We should look at it as an advantage. It might mean that we need higher and higher sample sizes. It might mean that we focus on finding different allele frequencies and section them out in different ways. But all in all, I think there's hope there, and we should, just like the overall genetics community, right, had to start by restricting analyses to white Europeans because they wanted to avoid confounding. Now there are chip arrays that are more representative of non-white populations. And the community in general wants to increase discovery by including multi-ancestral analyses. The same goes to say for admixed populations. And so I think that's still a road that we can go down. The problem of drug prices, though, I don't personally have a solution for. I mean, that is a huge health system problem that doesn't only affect gene therapies, but it is certainly a giant problem.
Keolu, I want to know, do you have ideas?
Yeah, I was going to flip that question back to you.
Choke ideas. But I think, one, with this team of people coming together, we will inevitably provide solutions for these things because we have to, because it just doesn't make financial sense. I think a lot of it lies in the way that we get in control and position of our data, because data is a resource, and then the way we build financial infrastructure around that so that it serves us, so that we're not outsourcing everything. You could imagine us starting our own series of NGOs, startups, pharmaceutical entities that serve our community's interests and prioritize our health and buying our land back and things like this. So very exciting future for us, but it's going to involve a lot of people. And I think this is the beginning of some really cool stuff. But we do have another question. It says, nice talks. Do you have any plan to conduct personalized medicine research based on the individual genome information, not just one or several genes of the Hawaiian population?
Do we have plans to offer personalized medicine? Well, not immediately. I think that beginning with some very key partnerships, we can begin to offer existing, very carefully screened genetic testing to our populations. But similar to the way that, the challenges that Leah faces when she offers genetic screening to our populations here, the diagnostic yield is probably going to begin as not as good in our populations. But it's a start if we can pair that process of offering precision medicine strategies and information with a research process. And I think that will be the way we start, and so that it can be both, increasing our research capacity while already offering some clinical information based on genetics. And then it will grow more and more accurate as we build a database for our populations.
Agreed.
Yeah. You want to add anything, Dr. Dowsett?
No, I mean, I think that's perfect. I completely, I'm on board with that. It's going to be an evolving type of science, but again, with the right community partnerships, anything's possible.
Oh, I just wanted to add too onto the idea of reducing the costs of many of these emerging drugs. I think that what has happened with that cystic fibrosis drug, the Vertex one, that's $300,000. It's interesting because the way it was developed was by recruiting patients that had cystic fibrosis and then kind of zeroing in on biomarkers or protein domains that allow you to develop a more advanced and effective version of the drug. So the drug could only become more efficient because of the participation of patients. And they should have reciprocated in some way. They should have created benefits or thought about benefit sharing models. So the new generation of pharmaceutical companies, a lot of them are thinking about this. They're thinking about benefit sharing and providing access points and intellectual property and at-cost drugs for people who participate in this, for people who will benefit from these drugs. So I think it's our kuleana to put more pressure on these companies to do that, because if everything is profit driven to the point where it's all about exponential growth but not efficacy, then we have a problem. And so just my 2 cents on that one. Did you guys want to say anything else before I do some more housekeeping? One thing, before everyone starts leaving, we will have another one of these seminars, and the next topic will be vaccine hesitancy. And it should be very fun. And obviously we're going to talk about the development of many of these new vaccines and how effective they are and how safe they are and how scared you should be, or should you be scared at all? So anything else you want to add?
No, this was fun. Thank you so much.
Mahalo to everyone for, asking great questions and for participating. Mahalo to Dr. Brown. Oh my gosh, blew me away. I really enjoyed to hear your talk. And again, Keolu.
Back at you, Leah.
Always a pleasure, Keolu.
Just mahalo everyone for joining us. Enjoy your weekend, whatever it is that you're doing, all over Hawaii, all over the world. If you're listening, thank you for our participants. Thank you to all of our sponsors and organizations that support these conversations, especially to JoAnn Tsark and Kauka Emmet Aluli.
Yeah.
We appreciate your aloha and love. And everyone, we'll tune in with you next time. Thank you so much. Mahalo nui.
Oh.
Ma'ana.
Oh.